Hi everyone, today we are going to go over our module 7. So we're going to start with amines. When we say amines, we are referring to any hydrogen or any nitrogen, that's bonded to any hydrogen. So the simplest amine will be ammonia, where everything that's connected to nitrogen is hydrogen, okay? So hydrogen here, hydrogen here, hydrogen here. Okay, so that's our ammonia. If any of those hydrogens are replaced with R group, remember R group is any compound that has a carbon, so carbon containing compounds, okay? So if there is only one carbon connected to the nitrogen, we call it primary amine. It's like the alcohols. When one carbon is attached to the OH containing carbon, we call it primary alcohol. Now it's primary amine. Amine is a nitrogen containing compound, nitrogen that is connected to hydrogens. Okay, so if nitrogen is connected to one carbon, that's primary amine. If it is two R or two carbon containing compound, that's secondary amine. If all of those hydrogens over here replaced with R group, then we call it tertiary amines. So when we look at this structure, what type of amine is this one? So when it's asking amine, we are going to find the nitrogen. This nitrogen is connected to one carbon here and another carbon here and a hydrogen here. So since there are two carbons, it's a secondary amine. Okay. So how are we naming the amines? Amine is a carbon chain that's connected to nitrogen. Okay, so we are going to count the number of carbons. 1, 2, 3, 4, 5, 6. So 6 carbon. I am writing my alkane name. If it is a 6 carbon, right, 6 carbon alkane name is hexane. So to be able to make it to amine, because it's connected to nitrogen, so we are going to drop this A and we are going to add amine. Okay, so it's a hexanamine. Okay. If there is anything that is substituent attached to any of the carbons, then we have to say where that substituent is located. That will tell you the location and name of the substituent. Okay, next group is amides. Amides are similar to many of the compounds that we talk about that has a carbonyl carbon, right? This is a carbon double bonded to oxygen. Carbonyl carbon, there's another carbon, there's another carbon. But next to carbonyl carbon, what makes amide is a nitrogen. Okay? So when we are naming the amides, we are going to count the carbons. 1, 2, 3 carbons. So 3 carbon alkane is, remember, we are naming always starting with the alkane name. Alkane name, 3 carbon alkane is propane, right? So we are dropping the E and we are going to add amide, propanamide. Okay, so 3 carbon and we're adding amide. What makes amide? Amide is the carbon double bonded to oxygen, carbonyl carbon, attached to nitrogen. And that nitrogen can be just attached to hydrogens, like in the case here, or it could be different R groups. Okay, and on this side of the carbon, there is another carbon containing compound or R group. Okay, so let's look and name one of the amides now. This is a carbonyl carbon. On one side of the carbonyl carbon, I have a nitrogen, so that makes it amide. First thing, when I name, I am going to say what is located on the nitrogen. On the previous one, oops. Okay, on the previous one, we had only hydrogen attached to a nitrogen, so we didn't say anything. On this one, we have one methyl group here, right? One carbon is methyl. The other carbon is also methyl because it's a one carbon molecule, methyl. So there are two methyls attached to the nitrogen. And the other part is 1, 2, 3 carbon. So first, we are going to say the N because it's attached to the nitrogen, name of the substituent. Then we are going to name the remaining part, prefix with anamide. Okay, so what is located on N? So I'm going to say N because this time I'm not saying the number. There is only one N and everything is attached to that N. That's how we name amides. N has a methyl, N has another methyl. So N has a methyl. I can say N-methyl because there are two of them. Or I can say N dimethyl. Okay, now we name this part. We're done with this part. Okay, and now the remaining part, we're going to count the carbons, 3 carbons, propane, amide. Okay, propanamide. So N-dimethyl propanamide. This could be very similar to your exam question or Cengage question, but it is not asking the name of the compound. It's asking what's the functional group in this compound. So in the exam, you're going to have a lot of those questions. They will ask you to recognize the functional group. So in this case, I am looking, it has a carbonyl carbon. Which groups has carbonyl carbon? I have the aldehydes, right? Aldehydes. I have the ketones. I have the acids, carboxylic acids, and I have the esters. And I also have the amides. Those are all carbonyl carbon containing compounds. So which one is this one? Next to the carbonyl carbon, I see nitrogen. So that makes it amide. Okay. What is the name for each of the following compounds? So let's look at this one. Yeah. I am looking at a carbonyl carbon. Next to the carbonyl carbon, I have a nitrogen, so I know this is an amide. First, I start with naming what is located on N. If N is just located connected to the H, that means you don't need to worry about it. Okay, so we don't need to say anything on this side. Now we have to name this part. What is this part? This part. So there is one, let's see. Okay, we have to worry about this part. How many carbon is here? 1, 2, 3, 4. So it's a 4 carbon molecule. 4 carbon molecule is butane, and because it's amide, butanamide. Okay, so we name this part. And now I have to say the name of the substituent. Where is my substituent? What is it? It's methyl. Where is it located? 1, 2, 3. So it's a 3. Carbon number 3 has a methyl. And the name of my compound is, the whole compound is butanamide. Okay, let's look at the next one. So I'm going to locate my carbonyl carbon. That's here. And it's located to nitrogen. So it's a amide. On that nitrogen, what is located on the nitrogen? So I'm only looking what is directly attached to the nitrogen. On this side, I have 1, 2 carbon. That makes it ethyl. On here, I have another ethyl, right? Excuse me. So nitrogen has one ethyl. Nitrogen has another ethyl. So we're going to say N has ethyl. N has ethyl. Since there are two of them, N diethyl. Okay, so we're done with this part. Now we have to worry about the other part. How many carbon is here? 1, 2, 3, 4. It's exactly the same as the upper molecule, except on this side this had only hydrogen, this has ethyl. Okay, so the remaining part we're going to name like the upper part. So it's a butanamide, but it also has a substituent. Where is my substituent? 1, 2, 3, 4. Carbon number 3. So it has a 3. My substituent is methyl because it's 1 carbon. Methyl because it's a 4 carbon molecule, butanamide. Okay, boiling and solubility, we're adding 2 more and then we are completing our whole chart. So we were having carboxylic acid as the highest boiling point, having compound or the high solubility. Now that we have amide is before the carboxylic acid. So amide, carboxylic acid, alcohol, ketone, aldehyde, amines are here, ester, ether, alkane. Okay, so this is our chart. Make sure you remember this chart. Okay, then we have the group of anhydrides, and the only questions that you will have with this, match which one is the anhydride. So you need to know this formula of the anhydride. You have 2 carbonyl carbon, O in between, R on both sides. Okay, so there is no other compound that will contain 2 carbonyl carbons. So if you see 2 carbonyl carbons on one molecule, that means it's an anhydride. Okay, now we're going to start talking about our carbohydrates, sugars, but before that I'm going to talk about concept chirality. Okay, so because those molecules are all in space, we are seeing them as like 2-dimensional when we draw them, but in reality they are 3-dimensional. And when you take one molecule, they can be present in mirror image of each other. So, and that determines their different characteristics. So it's important to know that what is exact structure of the molecule. So the term chirality or chiral molecule, we're talking about the chirality of the carbon here. When we say chiral carbon, remember carbon can make 4 bonds, right? And those 4 bonds, if each bond is connecting this carbon to a different molecule, okay, then we call it a chiral carbon. So 4 different groups are attached to the carbon. Then it is a chiral carbon. It's asymmetric and cannot be superimposed. Okay, so when you put chiral groups on top of each other, you cannot superimpose them. They are mirror image of each other. When we say achiral, at least 2 groups on the carbon are the same. So if I have a carbon, let's see if I have hydrogen here, let's say methyl here, ethyl here on this side, and on this side I have, okay, let's see, what do I have? I have CH2, CH3, okay. So on one side I have hydrogen, on the other side I have methyl, I have ethyl, I have propyl. So all 4 bonds are connecting this carbon to different molecules. So this carbon is a chiral carbon. On the other hand, let's say if this carbon now, if I have a hydrogen here, now this carbon has 2 hydrogen, 2 of the same group attached to this carbon, this is not a chiral carbon. Or if this is a CH3, because there are 2 CH3s on the same carbon, this is not a chiral carbon. Okay, for a chiral carbon, each bond that is connecting carbon to the other molecules should be connecting them to separate molecules, different molecules. Okay, so let's make an example. I have an example here. Decide if each of the following molecules is a chiral or achiral. As long as you can find one chiral compound, you can say that molecule is chiral. So you can choose any of the carbons and try to show that it's a chiral or not chiral. If I choose this first CH3, what does CH3 mean? Basically, C is here, it's connected to hydrogens, right? So this carbon is connected to 1, 2, 3 hydrogen. Remember, for a chiral molecule, it should be 4 different molecules attached to the carbon. If it is 2 or more, then we call it chiral. So this carbon is achiral because it's connected to 3 hydrogen. This carbon is also achiral because it's connected to 2 hydrogen. When you see CH3 and CH2, those are all achiral because the carbon is connected to at least 2 carbon or maybe more. So I can be hopeful about this carbon. So let's look at this carbon. On one side, it's connected to CH3. On the other side, basically, it's connected to hydrogen here, right? That hydrogen is here. On this side, it's connected to this 4 carbon. On this side, it's connected to this 2 carbon. So on all sides, this carbon is connected to different molecules. So it's a chiral molecule. So usually, when you are checking the chirality of the compound, so try to choose a carbon that it has a substituent, because the others, see this one already has 2 hydrogen. This has 3 hydrogen. So you can eliminate those ones. So I can focus on this carbon. On one side it's connected to CH3. On other side it's connected to CH3. So there are 2 CH3s in the same carbon. So this is achiral. I can come and look here. Okay. On one side is CH3. On the other side it's CH3. So there are two CH3s on the same carbon. That is achiral molecule. Again, let's look over here. 1 CH3, 1 CH3. There are 2 CH3s on one carbon. It's achiral molecule. Okay. So these are already achiral because they are at least connected to 2 hydrogens. Okay. Select the chiral atom. So one of your Cengage activities that you will be having those kind of molecules that you will look at the chirality. So if you look at here, you can at least find one of them. So you're going to be looking around the molecule to find one chiral molecule. On this side, it's connected to hydrogen. On that side, it's connected to OH. On this side, it's connected to carbon containing OH. And on this side, it's totally different. Okay, so this is a chiral molecule. Okay, now we can start talking about the carbohydrates. Okay, we all need to cut the carbohydrates, but not like this, right? And why the carbohydrates important? As a nurse, you will be hearing the sugar level of your patients. Diabetic is a common problem among us, like many of the people we know, or even ourselves in our families. It runs in my family, it runs in my husband's family, so it's our part of our daily life. So we are constantly measuring our blood sugar level. Okay, carbohydrates. When we talk about the carbohydrates, we are going to divide them into 3 big groups. We are going to talk about the monosaccharides, we're going to talk about the disaccharides, and the polysaccharides. So let's start with the monosaccharides. Monosaccharides, the one that we will be dealing with, the more common ones, there are 3 of them: glucose, fructose, and galactose. Okay, all of those molecules are 6 carbon molecules. They contain 6 carbon. Okay, and these are how they look. Probably you will not be able to recognize, but you need to recognize glucose is a hexagon, galactose is a hexagon, fructose is a pentagon. Okay. Okay, when 2 monosaccharides come together, they're connected by glycosidic bonds. So this is one monosaccharide, the other monosaccharide, there is a bond that connects them two, that's called the glycosidic bonds. So you can add the monosaccharides to each other to make disaccharides, or we will see polysaccharides. So the common disaccharides are maltose, lactose, and sucrose. Maltose is present in starch. Lactose is present in milk. Sucrose is present in fruits. Okay, so lactose. If you have, let's see, lactose, okay, you have a glucose plus galactose. Remember, this is a monosaccharide, this is a monosaccharide. They come together to form lactose. That's a milk sugar. Maltose, two glucose connected together through a glycosidic bond that makes it maltose, and that's present in starch. Sucrose, so now sucrose has a fructose, 5 pentagon. It's a 6 carbon, but still pentagon. The structure is different. And you have a glucose. So glucose and fructose makes it sucrose. Okay, so lactose, maltose, sucrose are disaccharides. Sucrose is present in fruits, maltose is present in starch, and lactose is present in milk. Okay, and then we have the polysaccharides. Examples of polysaccharides, we have starch. It's a storage molecule. If you have extra glucose in the plants, they store it as a starch. So we don't have starch in our bodies, we consume it, but we don't make starch. Starch comes from the plants. Glycogen, that's the storage form of glucose in animal cells. That's how we store glucose. And cellulose, that's also polysaccharide that's present in plants. It's plant cell wall. Okay, monosaccharides are connected together by glycosidic bonds. So between each monosaccharides, there's one glycosidic bond. And these bonds, when they are connected together, so you can consider this one as one monosaccharide, another monosaccharide, and between them is the glycosidic bonds. And these bonds can be either alpha or beta bonds. And polysaccharides are made out of, the ones that we're going to cover, all of those three are made of glucose. Okay, so the glycogen in here, it's a branched molecule, and the bond type between the glucose, they're all glucose polymers, glucose connected together to form polysaccharides. So the bond is alpha 1,4 glycosidic bonds. There is amylopectin, that's part of the starch, the branched part of the starch that has alpha 1,4 glycosidic bonds. And amylose is a linear molecule that's also part of the starch that also 1,4 glycosidic bonds. And cellulose is present in plant cell wall, and it's a linear beta 1,4 glycosidic bonds. Okay, so we said the glucose, fructose, and galactose, those are structures, either hexagon or pentagon. When you open those structures, we can write them as open structure. We don't have to draw them as a ring. We call it the Fisher projections. So when you open them, you will see the structures, and we will classify them as either aldose, ketose. So O's is for sugar. That tells you it's a sugar. Okay, it can be either aldehyde sugar, keto sugar. How do I know? Well, we already know what aldehyde and ketone looks like. Both of them will contain carbonyl carbon, right? This is a carbonyl carbon. If one side of the carbonyl carbon is attached to hydrogen, that's aldehyde. Okay, a carbonyl carbon, and this is attached to carbon. This part is already carbon. So this is R, right? C double bond O, R. And this is H. Okay, so this is aldehyde, this is ketone. So this is a ketone sugar, this is an aldehyde sugar. So we call it, this is a form of glucose, but when you're naming, it's then, or when you are saying the group that it belongs to, it's an aldehyde sugar, and it's a hexose because it contains 1, 2, 3, 4, 5, 6 carbon. And also fructose, 1, 2, 3, 4, 5, 6 carbon, it's hexose, but it is keto. Why is keto? Because carbonyl carbon is connected to a carbon, not the hydrogen. Okay, so monosaccharides are the simple sugars that they're not connected to other sugar. If it contains 3 carbon, 1, 2, 3, we call it triols. If it contains 4 carbon, that's a tetrose. 5 carbon, pentose, hexose, okay, and another hexose. So when you look at here, carbon attached to hydrogen, that's a aldo triose. This is also aldo tetrose, right? Tetrose tells you how many carbon is there. Aldo tells you whether it's an aldehyde type or ketone type. This is also aldopentose. Okay. This one, carbon is connected to carbon, so it is a keto hexose. This is an aldo hexose. Okay, so you will be given those structures and we're asked to classify it. So I can see CHO here, and the remaining is just the carbon containing molecule. So if I open this CHO, as soon as you see CHO, that's a aldehyde, because this is what it is. Okay, so because it's an aldo, 1, 2, 3, 4, 5, 6 carbon, aldo hexose. Okay, when you look at the Fischer projection of the molecule, you are going to look at the OH on the second carbon from the bottom. Okay, so you are going to start from the bottom and you are going to count the carbons. 1, 2, 3. So this is a 3 carbon molecule, and I'm focusing on the carbon above the bottom carbon. If the OH is located on the right, this is called the D form, derecha for Spanish for right. And if the OH is on the left, that's the L. So we call it L or D enantiomers. And it tells you where certain groups are located in space so that you can understand the characteristics of the molecules, because they will behave differently. Okay, so if the OH is on the left, we call it L. If the OH is on the right, we call it D form. And where are we looking? We are looking at the OH group that is attached to the second carbon to the last carbon. So when we are doing the Fischer projection, if that's, you draw a line on the backbone, this is the last carbon. So you are looking at the carbon before the last carbon, okay? And we are looking where the OH is located. And glyceraldehyde is the simplest sugar with 3 carbons. So, and you will see that name in the assignments too. So this is a D-glyceraldehyde because the OH is on the right. This is the L-glyceraldehyde because OH is on the left. Okay, so this is how much we're going to go into the carbohydrates. The next group is proteins. So proteins are another macromolecules that are very important in our body. It's everything that's done, all the jobs in our body are done by the proteins. So what are proteins? Proteins are molecules that are made out of amino acids. So remember, we talked about the amines, we talked about the acids. Now they're coming together to form our body parts. So this is the structure of the amino acids. And in the center, there is one carbon, that's the alpha carbon. Connected to the alpha carbon, we have COOH, that's a carboxylic acid group. And we also have the amine group, NHH. And this amine is connected to carbon, so that's a primary amine. So this is amine. Okay, that's acid, so that's why we call it amino acids. Okay, amino acids has carboxyl group, like we circled it, amino group, and there is one H group, that's also R group. So most of the amino acids, all of the amino acids will have the COH group and amine group. What is different between the amino acids are the R groups. So there are 20 amino acids in our cells that makes up our body, and they're all similar in that they have amine and acids, but their R group is different. So all of the amino acids except the proline contain at least one primary amine. So let's look at our amino acids. So if you look at the glycine, this is our alpha carbon. Connected to alpha carbon, we have COO. So this is a ion that it lost one of the hydrogen, so it becomes negatively charged. But normally there should be a hydrogen over here. Okay. So this is a carboxylic acid group. This is an amine group, and we have hydrogen attached to it. So this hydrogen is common to in all of them. Okay. This other hydrogen, okay, let's draw it. This is the R group for glycine. So each amino acid has a amine group, amine, okay, and they also have R groups. So R groups is different between all the amino acids. That makes them different amino acids. So glycine has a hydrogen as an R group, alanine has CH3, valine has isopropyl, and cysteine has CH2SH, proline has different. So what's important here, all of amino acids except proline will have primary amines. That's what the sentence was. Primary amines, that means, remember, amine groups are on this side of the molecule. This is our amine groups. Okay? And these amine groups are connected to one carbon on this side. So they're all primary amine. But when we look at the proline is connected to one carbon here, two carbon here. So proline is a secondary amine. All the others are primary amine. Okay, they're all primary amines. You don't need to remember the names or formulas of the amino acids. They will be given. In your Cengage exercise, you will have several questions from this chart. So it's going to ask what is the group that tyrosine belongs to. So tyrosine is a polar molecule. So there are non-polar amino acids. Glycine, alanine, valine, cysteine, proline, leucine, isoleucine, methionine, tryptophan, and phenylalanine. That's non-polar. Means they don't like water. And polar, serine, threonine, tyrosine, asparagine, glutamine, they like water. That's polar. There are charged amino acids. Some of them are basic. That will be positively charged. And some of them are acidic. They will be negatively charged. Okay, this chart will help you for the exercise, but you will not have the question in the exam from here. Okay. All amino acids found in proteins except glycine are in the form of L. Remember we talked about the L or D enantiomers? For the sugars, we looked at where the OH is located. For the amino acids, we are looking at the position of the NH2. If the NH2 is on the left, it's an L form. If it is on the right, that's a D form. So when we look at the serine, so we are looking at this NH2, it's on the left, so this is L. NH2 is on the right, that's D. In our bodies, we have amino acids in the L form, okay? And some bacteria might have on their D form. So let's look at this one. This is a glycine. That's the simplest amino acid. When you look at the glycine, this carbon has two hydrogen. So since it has two hydrogen, we cannot really talk about D or L form. It doesn't have a form. To be able to talk about the D or L form, the carbon has to be chiral carbon. So this is not a chiral carbon, that's an achiral carbon. And that's why there is no D or L form, okay? So you just need to know this because this will come up in the questions. All amino acids found in proteins except glycine are in the L form. Okay. Depending on what kind of environment the amino acids are, they're going to be charged differently. So at the pH, the acid level, below their isoelectric point. They will have a certain pH that they will ionize accordingly. So we will just look at this chart and we will know how they behave. At pH below their isoelectric point, amino acids are positively charged. So if the question says it's below their pi, isoelectric point, the amino acid will be positively charged. If it is above the pi, the amino acid will be negatively charged. Okay? So what is the net charge of leucine? That's an amino acid. In a solution, pH below its isoelectric point. Below isoelectric point, it's plus. Okay? So amino acids, yes, they are the building blocks of proteins. When they come together, they will make peptide bonds. So peptides are amino acids connected together. When they're connected, many of them together, we call it polypeptide, which is the other name for protein. Okay? So when one amino acid connects to another amino acid, it will form peptide bond. And that's called the primary structure. Amino acids are connected together by peptide bonds to make primary structure. If you consider this one amino acid, peptide bond, another amino acids. 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 So it will just keep going, and this is called primary structure. Okay, leucine is shown below. Suppose this amino acid was used to form protein sequence, and leucine will be here. Select any atom that will be a part of the protein backbone. So when we are looking at the protein backbone, this is the backbone that we are forming. Okay, this is the backbone. Any molecule that is in this line, it will form the backbone of the protein. So the R groups are not in the backbone. O's, double bond O's, are not in the backbone. Okay, what is in the backbone? The nitrogen is in the backbone. Alpha carbon is in the backbone. The carbon that is connected, double connected to O, is in the backbone. So if I look at this one now, this is my carbon that's connected to double bond O, so that's in my backbone. The carbon that's connecting to that carbon is also in the backbone, and the nitrogen is also in the backbone. So those three will be in the backbone. So you can just select any atom that will be a part of in the backbone. So you can choose any of those atoms that will be in the backbone. Okay, now you have the amino acids. When they're connected together through peptide bonds, it will form primary structure. Okay, so that's a primary structure. Within the backbone, then, those amino acids will bond together through hydrogen bond. So that's a kind of weak bond that can hold the molecules together. They will form hydrogen bonds between different amino acids to give them a secondary structure. Okay, so secondary structures are maintained by the hydrogen bonding between the backbone amide hydrogen and the carbonyl oxygen atoms. So when we look at here, those are the hydrogen bonds. So amide hydrogen is coming from this nitrogen, right? This hydrogen is bonding the carbonyl oxygens, this double bonded oxygen. Okay, so you can look at them. Double bonded oxygen is connecting to the hydrogen of the nitrogen. Okay, these are all your Cengage questions. You will not have that specific questions in the exam, but the secondary structure is when the amino acids within the protein primary structures, again, hydrogen bonded to one another to give it a secondary structure. And then we have the tertiary structure. So we had the primary structure, which is the amino acid sequence, amino acid chain. And then those amino acids are connected together through hydrogen bonds to give a secondary structure. Then we have the tertiary structure. They're held together by hydrogen bonds. Okay, the same thing as how The molecules within the primary structure are held together by hydrogen bonds to give this secondary structure. Then within the secondary structures, again, it can fold onto itself through hydrogen bonds, disulfide bonds, hydrophobic interactions, and salt bridges. It's the complete 3D structure of a polypeptide chain, okay? So it's not just a simple helix structure or little sheets, but it folds onto itself to give you a nice three-dimensional structure, and that's called a tertiary structure or 3D structure, and that's very important for the function of the protein. If several proteins that have their own tertiary structure come together and connect together, it's called a quaternary structure. Okay, so that means there are more than one chain of proteins. Okay, last group of molecules that we'll be discussing are the enzymes. Enzymes are actually a type of proteins, and their job is catalyst. That means they speed up the reaction by decreasing the activation energy. So normally two molecules are here. They try to go up here so they can go into the reaction and form the products. But for some reactions, this takes forever. So what's the function of the enzyme? That's the biological catalyst. They lower the activation energy so the reaction can happen fast. For sometimes the reaction can take weeks, but if you use the enzyme, it's microseconds. So we heavily depend on the enzymes in our body. Without enzymes, life is not possible because we need quick reactions to happen so we can sustain our life. All of the reactions in our body needs enzyme. And enzymes form enzyme substrate complex. So if you consider this one as an enzyme molecule, okay, it has a part that's what we call active site. Active site where the substrate will bind. When the substrate binds, it forms the enzyme substrate complex. And here the enzyme will do whatever it's supposed to on the substrate. Sometimes it cuts into two, sometimes it adds another group to change the characteristic of the substrate to turn into the product. Okay, so they have active site, and active site binds to the substrate. So active site is present on the enzyme. Okay, after the enzyme binds to the substrate, they form the product. And the enzyme is still there. Okay, nothing happens to the enzyme. Products are released from the active site, and now enzyme is ready to catalyze another reaction. It can go and bind to another substrate. So substrate is the molecule that binds to the enzyme to be changed. So it's a kind of like a reactant. Enzymes can be used many times. They don't wear, they don't get old. They function over and over again. They are not destroyed or used up during the reaction. They are not directly involved in the reaction. They are the helper. Their job is to bind to the substrate, do its job, and release the product. Just keep doing the same thing. Okay. The enzymes can be present as apoenzymes. So if we consider, let's look at here, we have the apoenzyme, that's the protein. But it is not functional by itself. It needs a helper, and we call it cofactor. That's a non-protein portion. When the apoenzyme binds to the cofactor, it will form haloenzyme or the whole enzyme. Haloenzyme is now ready to bind to the substrate. Substrate is the molecule that will bind to the enzyme, and it needs to be changed. A substance that functions to bind to an enzyme's active site and decrease its catalytic activity is known as inhibitor. Okay, so this is the enzyme's active site. Some molecules are binding to the enzyme and increasing its specificity so it can bind to the substrate. But there are also molecules that binds to the active site of the enzyme and blocks the substrate binding. So the substrate cannot bind anymore. So the activator molecules, they will, like cofactors here, it binds to the active site. Because active site like this, the substrate doesn't fit. See, the shape is different. But when the cofactor binds, it prepares the enzyme so the substrate can fit in. Okay, so those are the activators. We also have inhibitors. What does the inhibitor do? It binds to the enzyme. Now the active site is blocked. The substrate cannot bind to the enzyme anymore. So the reaction is blocked in the case of inhibitor. So there are different factors affecting the enzyme activity. pH is a very important factor. And most of the, so different enzymes has different pH requirements. So for this one, it requires pH 10. Okay, that's the optimum pH that it can function. And substrate concentration. So the more substrate you have, the more reaction you will have up to a certain point. Temperature will also affect the rate of the reaction. And enzyme concentration. So the more enzyme you use, the higher the rate of reaction. Okay, so the factors affecting the enzyme activity will be pH, temperature, enzyme concentration, and substrate concentration. Okay. So there are two types of theory that explain how the substrate binds to an enzyme. In the past, it was just a lock and key, meaning we have the enzyme here with an active site that matches exactly the substrate shape, and they will fit together, forming enzyme substrate complex. Now we have a little bit different explanation. We call it induced fit model. You have the substrate. The shape of the substrate is similar to the enzyme active site, but not really. It's not a real fit, but as the substrate comes closer to the enzyme changes its shape while it's binding and forming an enzyme substrate complex. So this is an induced fit model. So the binding of the substrate, or the initial binding of the substrate to the enzyme induces the change in the enzyme, and enzyme undergoes a conformational change to accommodate the substrate. Okay, there are classes of different enzymes doing all different kinds of job. There are oxidoreductase that catalyzes the oxidation reduction reactions. There are transferase that will perform the group transfer. So that means you are taking one group, let's say methyl group, from one molecule and transferring into other one. That's a transferase. Hydrolase, it's a cleavage enzyme. It cleaves the molecule into two different molecules with water. Lyase, breaking bond with elimination. So addition of a group to a double bond or removal of two groups to form a double bond is a job for lyase. Isomerase, they will make the stereoisomer switch. And the ligase form new bonds between the molecules. And again, you will have questions in Cengage activity, and you can come back and look at this chart when you need help. Okay, that's it.